For our first session, we have Nick Kooyers, who will be sharing information about the consequences of assisted gene flow on threatened annual monkeyflower populations. Nick?
Thank you, Holly, and thank you, Jeremy. Let me share my screen a second here. All right. So everybody looking at a pretty monkeyflower population? Yeah?
Yeah.
Okay, wonderful. Okay, so getting started here today. Before I start, I would like to, one, thank my lab because this is an entire lab project over 6 years. And most specifically, I'd like to acknowledge Donna Hendricks on this, this is her graduate work, is mostly what I'm presenting today. So I think synthesizing both the literature on model species responses to climate change and on some of the talks yesterday, we can safely say that herbaceous plants are strongly impacted by climate change. And what I mean by that is that at least in certain parts of the range, we are seeing maladaptation, or we're seeing declining populations in terms of population growth rates under 1. We're seeing that plasticity is normally necessary, but is not sufficient to actually recover any of that maladaptation or those declining population growth rates. And we're also seeing that species are dispersal limited and aren't able to disperse along with changing conditions. And what this means to me is we have to start considering alternatives for management strategies. And one of those major strategies is assisted gene flow, something that came up a lot yesterday. Assisted gene flow is the translocation of individuals within their species range to either facilitate adaptation or to add genetic variation to populations. And it can be illustrated really well with this buttonbush example over here. Say we have threatened populations in California. Well, if they're climate threatened populations, we may want to move individuals from hotter and drier populations, say these guys from Arizona, into those threatened populations in California to see if we can recover and get our populations with increasing growth rates again, or at least stable growth rates. And we can have these kind of expectations for assisted gene flow. So before assisted gene flow, we have these controlled populations and populations where we're going to do assisted gene flow into, which I'll abbreviate with AGF for the rest of this presentation. After assisted gene flow, we expect that over time, our assisted gene flow populations will have some recovery and do better in terms of relative fitness relative to our control populations. And if you look at a single time points in this and make a comparison with a box chart, what you can see is in control populations, you wouldn't necessarily see huge increases in between pre and post assisted gene flow because you're not doing anything to those populations. However, in the AGF modified populations, where we've now introduced new germplasm, we should both see increased fitness as well as greater standard deviation and greater variation in fitness in those populations, because you're introducing new alleles that both can create maladaptive combos, but also may create individuals that have higher fitness than the native populations that were there in the first place. Now, assisted gene flow is highly controversial in the conservation community. Normally, if you're looking at population growth rates, the population growth rate of stable being 1 over here before assisted gene flow. When you conduct assisted gene flow, depending on how many maladaptive alleles you have in a particular population that you're introducing, you typically think you're going to see a decline before you start seeing this pattern of evolutionary rescue occurring over time in these populations. So there are questions that exist. For instance, what if native distant hybrids that you're introducing are maladaptive? What if those distant alleles completely swamp out the native alleles and we lose most of the genetic variation that was present in that population? And what if distant alleles are only favored in some years, but not other years, and potentially creating problems in years where there are extreme events, for instance? Now, we don't have a lot of systems where we have conducted assisted gene flow. Most of our systems are theoretical or are laboratory-based experimental evolution systems, or alternatively, are systems that are of high conservation interest where we see declining populations. And such, we can't normally do proper controls or experimental design to test how assisted gene flow is actually working. So systems like sagebrush, west, elkhorn corals, several different tree species, including redwoods, have some assisted gene flow going on within them. So my questions for assisted gene flow are largely thinking about how assisted gene flow can change the mean fitness and the variation in fitness over time, and if introducing particular growth stages or life history stages is actually going to be more effective than others. How do these AGF responses vary across populations and across different years, especially across extreme events? And then finally, can we use genetics to track those non-native alleles intergressing within populations and determine how much integration is occurring, as well as if we're swamping out any of those native alleles or native genetic variation? Basically, can we do AGF effectively within threatened populations? The system that we're going to be talking about today is Mimulus guttatus. It's been the system for evolutionary genetics and ecological genetics for a substantial period of years, almost 100 years now. Many people study it for those different fields because it has a really wide geographic range that spans from Mexico to Alaska and from sea level up to 11,000 feet. It has substantial genetic and phenotypic variation in those different populations that allow it to adapt to those different areas. We have a number of really good reference genomes in guttatus. We also have thousands of resequenced genomes now. You're going to see about 850 later on today. There's a developed toolbox and a long history of ecological experiments dating back to Clausen, Keck, and Heisey. And it's just a really nice system for doing field experiments, for doing lab experiments, has a rapid generation time, and just an incredible diversity. I love this species. One thing that you need to know about monkey flowers here is that their growing season is largely limited by water availability, especially the annual populations that I study. The growing season starts when we have winter precipitation, either in terms of rainfall or snow, and it ends in terms of a terminal drought in the summer in these Mediterranean climates. So it gets hot and dry, and essentially these populations that I'm looking at have ephemeral water supplies that don't allow them to make it to the next year. So throughout this range, they have really high diversity in when the growing season starts and how long the growing season proceeds. This widespread species with a ton of genetic variation is something that you would not expect to have the struggle with changing climates. It should be able to complete evolutionary rescue itself, or you think it would. But there are some areas in the Central Oregon Cascades where this species has the shortest growing season. The growing season lasts there for only a month to about 7 weeks every year, right? And in these populations, this is the populations that we think may be threatened by changing climates. So way back nearly a decade ago now, we did these provenance experiments where we planted individuals in low and high elevation populations. We planted individuals from California low and high elevation populations that we think better match the contemporary conditions in some of those Oregon populations in terms of growing season. And we also planted our native populations in each one of these gardens too. And we came up with this kind of beautiful signature of adaptation lag. When I say beautiful, that should probably be in quotation marks. In the low elevation site, we see that low elevation California plants do better than the native population. In the high elevation sites, we see that high elevation California plants do better than the Oregon native population there too. This is kind of your classic signature of climate-induced maladaptation or an adaptation lag. And this suggests that these populations, and particularly the ones at high elevation, are struggling to keep up with changing conditions. Thus, while the species is not threatened at a range-wide scale, these individual populations may be threatened. And this may be a canary in the coal mine for thinking about other species that are more endemic and more narrowly distributed. Okay, so this is where I started off my faculty position 6 years ago here at University of Louisiana. And I decided to take a playbook out of David Inouye and all these other really great plant biologists looking at changing climates and start some long-term observational experiments, especially in these Oregon Cascades populations. So I started tracking 12 different populations here, where every year I would go out and I would run transects, and we'd pick up 50 to 100 plants along each transect. We count the number of flowers and number of seeds that each one of these plants had. We take those seeds back to the greenhouse, plant them in common gardens so we could survey morphology, phenology, and physiological phenotypes. And then we take data for genetics. So we do whole genome sequencing on 24 individuals per population per year. Well, it only took a couple of years here before we figured out that there were some that these populations were declining faster than we thought. In fact, we saw extirpation of one of these populations in the first year. It eventually came back through the seed bank, but this suggested that we needed to start doing something about this in these populations. And this had a perfect design really for a local landscape scale assisted gene flow experiment. So we took those 12 populations and we divided them into 4 groups of very similar populations to one another. In each of those 3 populations, so in each of those 4 different groups, one of the populations was a control where we just threw out 200 native seeds into that population. One of the populations, we introduced seeds from California, and the specific lines that we introduced were a low and a high elevation population that were crossed together into F1s, and we threw those F1s out there. We did this because in different years, each of these populations was favored in a different year that we did previous provenance studies. And third, for the third population, we germinated seedlings and planted those in our populations. So notice we only planted 20 of these seedlings per population for in those seedling transplants. So it looks something like this. In 2018, 19, and 20, these are all baseline years pre-assisted gene flow. We did our assisted gene flow event in 2021, and thus our fitness in 2021, 2, and 23 are our post-assisted gene flow years. Now, within 3 years, we'd expect relatively limited changes because this is the first generations. Theoretical models suggest that we shouldn't see very much happening in these first years as alleles are just starting to integress into these particular populations. But what we would expect to see or hope to see would be that in our control populations, we don't see very much difference between pre and post generations. In our assisted gene flow populations, we see some increase in fitness relative both to the pre-assisted gene flow years and relative to the post-assisted gene flow years in those control populations. So without further ado, I'll give you the results. Okay, we were surprised. So overall, when we started running these models, it looks like assisted gene flow certainly has some impact on fitness within these different populations. Specifically, if we're looking at floral abundance, we can see when we introduce seedlings, we saw about a 20% increase in number of flowers in those particular populations. And we saw about half that increase in the seedling populations relative to both the control and to their control populations in those same experimental groups. Now, we also saw when we looked at variation, that variation was higher in these populations where we had either introduced seeds or seedlings in terms of the standard deviation in floral abundance. This wasn't significant, but this was in the direction that we would have expected from introducing a bunch of new alleles within these populations. Now, looking at a second metric, if we look at seed production, indeed, we saw on average, we increased seed production again in the seeded populations, but not necessarily in the seedling populations. And this was not a significant effect relative to our control populations either here, suggesting that our effects are in the same direction, but we're really increasing flower numbers, but not necessarily the number of seeds that are contained in each one of those flowers. And same thing for standard deviation. We really don't see the same effects for standard deviation for seed production that we saw for flower production. So in broad terms, our results here suggest that assisted gene flow is effective in the populations where we seeded in a bunch of California individuals, specifically for number of flowers. So probably increasing pollination. Those number of flowers are going to result in increased pollination of different individuals in that population. We're going to have increases in some of those alleles coming from those California populations. So this is specifically in the populations that we introduce seeds, but not seedlings. So why not seedling populations? Well, the reason is we introduced in 2021. And in 2021, there happened to be a really strong heat dome event that produced some of the hottest temperatures ever seen in Oregon and some of the hottest temperatures on record within our field sites. So this is at 4,500 feet, and it's over 100 degrees Fahrenheit at the end of June here. This is our populations beforehand. Look at all the yellow, that's all monkeyflowers. This is 3 days afterwards. Those populations are toasted. These are some of our transplants right before this heat wave event. These are our transplants that are toasted after our heat wave event. And we had about 77% mortality to our translocated CA seedlings prior to reproduction. So they clearly were not introducing alleles within that population. And on average, only the surviving plants only produced a single flower most of the time. But what you don't realize is in those populations too, the natural populations only had far greater mortality than 77% at the same time. So our transplants actually did relatively well compared to the natural population. Okay, so did all the population groups respond the same way across all 3 years? Well, I just told you there was a gigantic heat dome event in one of the years, so that is definitely not what happened here. And we see some of the standard, when we see some of the differences in that seed production and looking at the amount of variation in all of our fitness metrics, it's clear that something's going on where certain site groups have differences in fitness in some years, but not others. And that's exactly what we see. In one of our site groups, the lowest elevation one, we see a strong effect of assisted migration, particularly in the seeds population, but also in the seedlings population, increase relative to pre-assisted gene flow and towards the control populations. In some we see weaker effects that may be driven by the control population having far less fitness in the years post-assisted gene flow. In others, though, we either see less effect or no effect or maladaptive effects. So here we see the control population has increased post-assisted gene flow, but all of the plants all the populations where we introduced either seeds or seedling into did not do as well relative to the control populations. In others where we see nothing. So there's a lot of variation between those 4 different replicates of assisted gene flow that we conducted. There was also a lot of temporal heterogeneity in these responses to assisted gene flow. So in 2022, we saw strong increases relative to pre-assisted gene flow years in all the populations, but higher in the seeded populations than in the control population. However, in 2023, we see that these populations did more poorly than they did in our baseline data. And we see just small increases relative in seeds to control populations essentially too. So our assisted gene flow is differing between years as well, depending on the yearly conditions within these different sites. So there are clear population by year interactions that are going to be important if we start thinking about how we want to manage these populations and using assisted gene flow as a conservation strategy. So given this, you might ask me, how sure are you that assisted gene flow is actually causing the fitness changes within these different populations? It's a question that I asked too. We only have 4 replicates, and each one of these kind of depends on the control population and how it did pre versus post-assisted gene flow. So we decided to use population genomics to validate our results and try to figure out how many California alleles are actually introgressing here, and if we see more in the populations that we either seeded or planted seedlings into. So you remember back that we did whole genome sequencing to sequence 24 individuals per population per year. We ran this through kind of your standard alignment, calling variants, and filtering pipelines for SNPs. And we ended up with about 15 lines per population per year with a mean coverage depth of about 11x and a mean error rate that's pretty low. So this is fit pretty well to what we thought was going to occur here. So in terms of total number of samples that we had pre and post assisted gene flow together, we had 872 samples, which we had whole genome sequences for. 24 of those lines came from the California populations, either the Bell parent line or the F1 lines. We had 481 lines that we took from pre-assisted gene flow from those 2018 through 2020 populations. And then we had about 367 that we took post-assisted gene flow. This is only 2021 and 2022. We're still working on the 2023 data. We're just getting it back now. When we call SNPs in all of these samples together, we get about 18 million SNPs, which is about what we'd expect because this is a highly diverse species. And when we look at how many of those SNPs are private to our California populations, we see about 160,000 across the genome are private to those California populations, just under 1% essentially. This is about what we'd expect. So if we're looking at this, we can ask, do we see more introgression into those seeded or seedling populations relative to our control populations? And here's what we see. So we see that in our seedling and seedling populations, we see relatively a higher number of introgressed California private alleles. We see a relatively low percentage of our California private alleles have actually introgressed. What's pretty crazy about this too is that you see in that control population, we see some of those California private alleles too. And one of the reasons that we might see this in these control populations is because we didn't do the greatest job beforehand of figuring out the standing genetic variation in each one of those populations. So some of those alleles are probably standing variants within those populations already that we inaccurately called as private alleles to California. But I have no doubt that we have some intergression between these populations because of pollinators. Now, across the genome, we see this intergression spans genome-wide. But kind of the cool part is it differs between different generations. This isn't exactly what we'd expected to happen. So the 2001, when we have really low frequencies of California private alleles, most of the time in 2022, we didn't see those same alleles in 2022. And when we saw some new alleles appearing in 2022 that we didn't see in 2021. This tells me right here that a single introduction event, we're going to have seeds that are germinating in those seeded populations over multiple different years from that particular seed bank. And we might be losing a lot of those alleles fairly quickly in the population because they're at really low frequencies. So we either might not be detecting them in the second years, or they might be lost through genetic drift. Okay, so back to the main question here. Can we find populations where we see both increases in fitness and increased integration from California alleles, the intended effects? Do we see populations where we have declines in fitness with assisted gene flow, but we do see integration of those alleles from California? Or do we just see there's changes in fitness with no integration whatsoever in those different site groups? So breaking this up between site groups, we certainly do see some in that first category where we see both increases in fitness and increases in integration of those private alleles between populations. We see others where we see maladaptation, where we see definitely that we have integration of private alleles, but we also have declines in fitness here, suggesting there's been maladaptation these first couple of generations, more of our standard expectation for assisted gene flow. And we see others that fit into these categories, where it's clear that the control population does not fit very well, is not a good comparison for those assisted gene flow populations where we had translocations. So in general, we find some evidence for really rapid success of assisted gene flow. We find others where we see that maladaptive effects in those first generations. We see better results in the populations we seeded versus the ones that had seedlings. These are incredibly heterogeneous responses across time and across different populations, likely because we introduced relatively few alleles. We have integration occurring genome-wide, but the frequencies are still quite low in the populations, and our next steps are largely determining the extent of that integration and the ecological mechanisms. I added this slide for Holly because this has been kind of a key here too. So takeaways for management here. Our seeded populations, we introduced about 5% of the population numbers for what we thought. The seedlings, far less. And we saw the introduction of seed work much better than seedlings. That might not have happened if we hadn't planted in a heat dome year and if we had introduced more plants there. We need to quantify these responses through time, because I think extreme events are going to have really significant impacts here. And that might be why we see more rapid increases in fitness in some of those populations. And finally, we still need to have a lot of effort put into the best practices for choosing the germplasm that's necessary. Which populations do we choose? How many populations do we choose? Do we just throw in distant seed? Do we throw in hybrids between native and distant seed? Or do we do distant hybrids like we did here? All still open questions that I have thoughts, but not for today. So thank you to everyone who has participated in this project, NSF funding, the Forest Service, who has been great with permits, and specifically Donna and Courtney for spending a lot of time on this experiment. And I'll take any questions in the chat because I'm right at my time right now. So thank you guys.